Welcome to the problem-solving set on mutation types and causes. Question 1. Arrange the following statements to the correct order of events of excision repair. So remember that excision repair is one of the DNA repair systems where a mismatched and modified base is identified and then nucleotides are taken out and correctly matched base pairs are put in. So let's run through these 4 statements. If we look through, first the mismatch has to be identified, right? So that would be 2 here. A mismatch base pairing is identified. So that's the first step. Then if you look through, the next step would be that a stretch of around 12 nucleotides gets excised out by an endonuclease. So that is step 2. Now once those 12 nucleotides are taken out, DNA polymerase comes back and adds nucleotides according to the correct base pair rules. So that is statement 1. And then finally, ligase comes around and closes the break between the old nucleotides that were there and correctly base paired already and the new 12 nucleotides that are put in. So ligase, that's statement 3. And this is the order of events from beginning to end. And this is only, remember, one type of DNA repair systems. There are other repair systems that remove only the one damaged base or mismatched pair, but this one that we're talking about here removes the 12 nucleotides. Problem 2. For the following sets of DNA sequences, describe the mutation that changed the first sequence to the second. So in order to answer this question, we're going to compare and contrast the 2 sequences given on right to left. For A, let's look through. ATGC is the first 4 on the left side, and on the right side, which is the second sequence, it's ATGG. So you see that this C was changed to a G. And then if you continue, TA, they're both TA at the end. So the only mutation in here was from a G to a C. Now because this was one base, we're going to call this a point mutation. And it's a C to a G. Now remember that C is a pyrimidine, and G, guanine, is a purine. So when you change from a pyrimidine to a purine, or from a purine to a pyrimidine, this is called a transversion mutation. Okay, so that's how you describe the mutation in part A. In part B, we'll say it's AT on the left and then AC on the right. And if you continue, both of them are GGTA. So the difference here is changing from a T to a C. Again, this is changing only one base, so this is a point mutation. And here, you're changing a T to a C, which are both pyrimidines. Now, when you change between a pyrimidine and a pyrimidine, or between a purine and a purine, this is called a transition mutation. Okay, and finally for C, the left-hand sequence is ATGGCT, and the right-hand sequence is only ATGT. So we can see here that these 2 bases are missing, right? They would be put in here, so that you have ATGT on the left, which would match ATGT on the right. So when you're missing 2 bases and they're just completely deleted, we're going to call that a frame shift mutation. And this is the deletion of 2 base pairs. Problem 3. Deamination of adenine converts it to hypoxanthine, a base that behaves like guanine with respect to complementary base pairing. For the following DNA sequence, determine the sequence of the resulting mutant DNA molecule post-deamination and describe the observed mutation after 1 round of DNA replication. So I don't think you really need to know this, but deamination would be a removal of an amine group, which is a nitrogen-containing group, so we'll just say removal of N. And that converts adenine to hypoxanthine. Hypoxanthine behaves like guanine, so we could say it's basically guanine. So it will complementary base pair with cytosine. So when you deaminate, this is only affecting the adenine, right? So when this piece of DNA is going through DNA replication, this 5' to 3' template will go off, and it won't be affected by this mutation. So it'll still be GCT CG 3', and it'll pair 3' CG A G C. So this is normal, and it's not affected by the mutation. However, this 3' to 5' template, this will go off, and remember now, this adenine is now deaminated, so it's acting as if it's a G, and therefore it'll base pair, so it'll be G, C, 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 G. Okay? So now it's asking us to describe the observed mutation. So what actually changed? Remember that although it's acting like a G, it didn't really change it into a guanine. However, this T right here did actually change to a C after replication. So if it's a T to a C, that's a pyrimidine to a pyrimidine, which is a point mutation. And this is known as transition. Okay? And then it's asking us also to determine the sequence of the resulting mutant DNA molecule. So in order for the mutation to fully change everything, this would have to go through another round of DNA replication, where this 5' template be 5' G, C, 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 G, 3', and it will pair with 3' C, G, an actual G now, another G, and a C. So now, after a full round of DNA replication where both templates are used to make a new strand, that original T is changed to a C, and this original A is changed to a G. So this is the DNA molecule. And remember, it's still a point mutation. And if you want to double check that, you can recognize that that A from the beginning is now a G, and an A to a G is a purine to a purine, which again is a transition mutation. Problem 4. The DNA sequence of a gene from 3 different mutants is given below. Use the information to determine the wild-type gene sequence. So in order to find the wild-type gene sequence, we're going to compare the sequence from all 3 mutants, because remember, these are from the same gene, it's just different mutations. So at the first base position, we see that all mutants have guanine. So therefore, the wild-type gene must be G at the first base position. For the second position, 2 of the mutants have C, whereas 1 of them have G. Now for the wild type, we're going to assume that base C is actually next, and that G is mutation in mutant 2 only. For the next base, they're all C, then they're all A, T, C, A. Now here, 2 of them are T's, 1 of them is A. So again, we're going to assume that that one variation is from a mutation in mutant 1, and that the wild-type gene has a T at this base position. Then they're T, T, A, C. Now here 2 are C's, and 1 is G, so we're going to assume that G is from a mutation. So it's C there in the wild type. Then they're all G, C, A. So as you can see, these 3 mutants only have 1 mutation each. So in mutant 1, they have a mutant A right here. In mutant 2, they have a mutant guanine right there. And in mutant 3, they have a guanine right there. So all of them have only 1 base mutated in their gene sequence. So we're going to say that they're all point mutations. Now it doesn't ask for us to do this, but we might as well go through and say what type of mutation each of these mutants have. So for mutant 1, in the wild-type gene at that base position, the wild-type gene had T, and now this mutant has A. So it went from a T to an A. Now that is a pyrimidine to a purine. and that is a transversion. For mutant 2, normally the gene sequence has a C there, but it's mutated to a G. Again, that's a pyrimidine to a purine, which is transversion again. And for mutant 3, they have a G there when there used to be a C in the wild type, so it's the same as mutant 2, transversion. It was just at a different base position. So all 3 mutants had a point mutation that was a transversion, and they actually all went from a pyrimidine to a purine. And just for your knowledge, remember that a transition would be a pyrimidine to a pyrimidine, like a C to a T, or a purine to a purine, like an A to a G. So this is just a side note. Question 5. Achondroplasia is a very rare autosomal dominant human condition that results in a dwarfed body size. A large hospital kept track of babies displaying this trait over several years and found that 26 babies were affected out of 100,000 births. Out of the 26 dwarfed births, 6 of them had a dwarfed parent. What is the apparent mutation rate of an achondroplasia gene in this region? Is it unusually high or low? So it's saying that out of the 26 births that had this condition, 6 of them had a dwarf parent. Now, because this is a dominant human condition, that means that 26 minus 6, or 20 of these births, happened through mutation. And that's because they had no family history. Okay? So the total number of births that happened at this hospital over the years was 100,000 births. Now with these 100,000 births, that means that there was 200,000 parents, right? So that means that there was 200,000 germlines that could have gone through this mutation to give the dwarf parents, the dwarf babies. However, only 20 of them actually went through mutation. So to get the mutation rate, we're going to say that 20 mutants out of the possible 200,000 that could have gone through mutation and that's the mutation rate, so we want to do 20 divided by 200,000, and that gives 1 times 10 to the negative 4th. Okay, so this is the mutation rate. Now, it didn't tell you in this problem what a normal human mutation rate is. So if you had to look it up in your textbook, or if not, I'll tell you right now, the average human mutation rate is actually 2 times 10 to the negative 6. So if you see, 1 times 10 to the negative 4th is higher than this. So this mutation for this dwarfed body size is actually unusually high. Okay, so say it's high, and the rate is 1 times 10 to the negative 4th mutations. Problem 4. A researcher performed the AIM test on 4 different substances to see whether any of them are mutagenic. For each substance, she incubated both with His minus bacteria alone and also with His minus bacteria with mammalian liver enzymes. Based on her data presented below, determine if each of the following statements are true or false. So remember just some quick facts about the AIMS test. So the AIMS test is for mutagenicity, right? And basically they take His minus bacteria mixed with whatever mutagen you think causes mutation. Now His minus bacteria is auxotrophic for histidine, so it cannot grow on media that doesn't have histidine. If whatever mutagen you're using actually causes mutations, the theory is that it will be able to grow on histidine lacking media, which means it would be reverted to histidine plus. Now so that's just for the histidine minus bacteria. When they mix the histidine minus bacteria with the liver enzymes, that's supposed to resemble the system of mammalian a mammalian body or an organism, and that's to see if any of these things would be mutagenic in humans. So that's why they mix it with the liver enzymes. Now, to determine the data, what you would have to do is see the number of His plus colonies or reversion colonies that went through mutation. If you see a lot of them, that means that the mutagen you added was actually mutagenic, and it could also be carcinogenic in humans. Okay, so now let's look through the statements. Statement A, substance 2 is unlikely to be mutagenic in mammals. So if we look across at substance 2, it gave no reversions to just the His minus bacteria, and even when it was added to liver enzymes, it had no mutant reversions. So therefore, it is unlikely to be mutagenic in mammals, so that's true. Okay, B. Substance 1 and 4 are likely to be mutagenic in mammals. So let's look at 1. Again, we're looking, because they're asking about the mammals, we're going to look at the bacteria that were added with liver enzymes. So for substance 1, this gave 0 revertants, and substance 4 gave 136 revertants. So with this data, we would say that substance 1 isn't going to be mutagenic in mammals. However, substance 4 seems to be highly mutagenic in mammals. So for B, this is false, and only substance 4 would be mutagenic in mammals. Statement C. Mammalian liver enzymes convert substance 4 to a compound that is likely to be mutagenic in mammals. So for this, you want to compare the His minus bacteria only with the His minus bacteria added with the mammalian liver enzymes. So if you see, when you just put substance 4 with the bacteria, you don't see any revertant colonies, so it's not mutagenic. However, when you add the liver enzymes, you see 136 revertant colonies, which means that you want to conclude that the liver enzymes are actually converting it and doing something to it to make it mutagenic. So for C, we're going to say true, that mammalian liver enzymes do seem to convert this substance into something that's mutagenic. Okay, statement D. Substances 3 and 4 are likely to be mutagenic in mammals. So if we see 3 and 4, again, we're looking at the liver enzyme column, and we see that they both produce a decent amount of revertants. So this is true. They're both likely to be mutagenic in mammals. And finally, statement E, substances 2 and 4 are mutagenic in bacterial cells. So we're going to look at 2. There are no revertant colonies when you're just looking at bacteria alone, and the same goes for substance 4. So when you don't see any revertant colonies, that means it isn't mutagenic to bacteria. So this is false. Neither are mutagenic to bacteria. Now this might be a little confusing because substance 4 does give revertant colonies, but remember that's only after you add the liver enzymes. And of course, bacteria are just single-cell organisms, and they don't have a liver, so they don't have these liver enzymes to make it mutagenic. Okay, so this goes over the Ames test. It's very important to see what chemicals or anything can become mutagenic and maybe carcinogenic in humans. Problem 7 is a fairly long problem about a cross between fruit flies. So what I'm going to do first, before we look at any specific genes and what's happening, I'm going to do the cross, and I'll explain to you how I'm going to do it along the way. So the first statement says that a wild-type female fruit fly was mated to a wild-type male that had been exposed to a high dose of X-rays. So let's do this parental cross. So because they're wild-type, I'm not going to draw any genes on it. So we have a female. So remember that they have 2 X chromosomes. And we're crossing it with a wild-type male that had been exposed to a high dose of X-rays. So, of course, the male is going to have a Y chromosome, but to differentiate the X chromosomes to say that he was exposed to X-rays, I'm going to draw it as a squiggly line, okay? So that is his X chromosome. So it says one of the resulting female offspring was then mated to a male that had 5 recessive markers on the X chromosome. So out of these F1 offspring, one of the females, so it's going to have 1 normal X from its mother, and the irradiated X from its father. And now we're crossing that with a fruit fly male, so it has a Y chromosome, and it has 5 recessive markers on the X chromosome in this order. So it has O, TW, TR, FU, VE. Okay? Now it's saying the progeny of this second cross included half the amount of males as females. So the F2 generation had twice the number of females than it did males. And if we want to just draw the chromosomes, remember that all the females are going to get the X chromosome from their father. So they're going to have O, TW, TR, FU, VE, and the other chromosome from their mother. So one of them will get, well, half of the daughters will get the normal X chromosome, and the other half, remember, will get the recessive markered one from their father, and the irradiated X chromosome from their mother. And here the males always get the X chromosome from their, the X chromosome from their mother, and the Y chromosome from their father. Okay. However, it's saying that there's twice as many females as there are males. So we're going to assume that one of these male options actually die off, and because of the irradiation through X-rays, irradiation can cause mutations. We're going to assume that these males didn't make it. They had lethal genes, okay? So let's see, let's continue. It says all of the males and half of the females were phenotypically wild type. So all the males are phenotypically wild type. That makes sense for the males because the only way to get phenotypically recessive would be to get the X chromosome from the male father, and obviously males don't get the X chromosome from their father. So this phenotypically wild type. And it's saying half of the females were phenotypically wild type, but the other half of females exhibited the TR and FU phenotypes. So if this X chromosome is intact and it has all the wild type dominant alleles, we're going to say that these females were wild type. And these females that had the irradiated X chromosome, they exhibit the TR and FU phenotypes. So they are TR, FU phenotype. Okay. So now we can answer the questions. Question A says, what did the X-ray exposure do to the original irradiated male? Well, let's see. Let's look at this female right here. She's exhibiting a TR-FU phenotype. Now, if the only way that she would exhibit this is that if the two genes, two alleles on this chromosome were either recessive or if they were missing. Because on this, you have this TR and this FU are the recessive alleles. However, if these were dominant, then they wouldn't be this phenotype. So we're going to assume that the irradiation actually deleted these two genes. So this is a deleted region. And if we assume that that was a deleted region, that actually makes sense that these males right here were lethal because let's say that a deletion causes inviable offspring. Okay? So the original X-ray exposure that happened to this X chromosome deleted, so we'll say A, the X-ray deleted the TR plus FU plus region. Now remember it's plus or wild type because this original male was wild type. So that answers question A. Now for question B, it says draw the X chromosome pair present in the phenotypically TR-FU females. So this is down here. So the chromosomes, I'll erase that up there. And we can answer B down here. So these TR-FU females, they have one chromosome that had all recessive alleles from their father. So it's O, TW, TR, FU, VE. And the other chromosome that they had was from the irradiated original male. And that had the deleted region of TR-FU. So here, because there's deleted regions, it's going to be shorter. And it has O plus for wild type, TW plus for wild type, and VE plus for wild type. Because remember that TR-FU region was actually deleted, so this extra X chromosome that they have is going to be shorter. Okay? So that completes this question, and I'll see you at the next problem solving session.